Welcome back to part 2 of chapter 4. In this video, we're going to be talking about the details of eukaryotic cells, including both animal and plant cells, and what they contain. What are the different structures that make up their organelles, and what are their organelles' functions? Looking at this eukaryotic cell, we can already tell that it's much more complex than the prokaryotic cell, bigger, and we have many more structures. One of those most prominent is probably this nucleus, this membrane-bound nucleus, which contains the DNA. We've got also many different types of membrane-bound structures shown throughout the picture. And these include endoplasmic reticula, we have mitochondria, and we're going to go through each of these throughout this second video. We have two major types of eukaryotic cells we're going to be looking at in this chapter. The one on the left is an animal cell, and the one on the right is a plant cell. And right away, what stands out for me at least is the rigidity, the square-like structure of the plant cell due to the presence of a cell wall. So it has a thick cell wall outside of the cell membrane, in contrast to the animal cell that has no cell wall, and so it has a more globular shape. I also see this giant vacuole in the middle of the cell. We'll talk about that in a bit. As I mentioned earlier, all cells have a plasma membrane, but eukaryotes have pretty complex membranes with different things embedded within the membrane. All membranes are made of a phospholipid bilayer that we saw before. Remember, a phospholipid looks like this. We have a water-loving hydrophilic or polar head and two water-fearing tails. So I have a bunch of them facing the outside of the cell, if this was the outside environment. But I also have another layer facing the inside, and it looks like a mirror image. So water-loving is facing over here outside of the cell, but the water-loving layer is also facing the inside of the cell. And then in between, we have the hydrophobic fatty acid tails. So most things actually cannot pass because of that fatty acid tail layer. It's hard for things to pass through if they are charged or polar, for example, since they're repelled by this non-polar layer. So in order to get into or out of the cell, we often have protein channels that allow these water-loving things into or out of the cell. We'll see that there are other things attached to the membrane as well. Glycoproteins are really sugar-protein complexes. Glycolipids look like a sugar-fat complex or sugar-lipid complex. And then some cells in our body even have structures that are extensions of the membrane, like this one shown here. This is what we see in our intestines. Instead of just having a normal cell membrane, they want to have a greater surface area to move things into the cell. So they've actually invaginated the cell membrane several times to increase that surface area. Everything inside of the cell, between the nucleus and the cell membrane, so all of this area between the nucleus and the cell membrane, is known as the cytoplasm. And this includes the liquid portion, the cytosol, as well as all of the organelles within the cytosol. So these two together make up the cytoplasm. Most of it is made of water, but it is gel-like or semi-solid because of all of the different proteins embedded within the cytoplasm. So the nucleus is usually the biggest structure inside the eukaryotic cell. We usually find it near the center of the cell. There's usually only one in each cell, but that's not always true. For example, in humans, our skeletal muscle cells are multinucleated. We have more than one nucleus, and the plural form of nucleus is nuclei. If I look carefully at the nucleus over here, I can see several different components of the nucleus. Inside, we have this string-like material, that's our DNA, in the form of chromatin. Chromatin, I have it here. That's just DNA wrapped around proteins. The gel-like layer inside the whole nucleus is called the nucleoplasm, kind of this like the cytoplasm was the inside of the cell. The nucleoplasm is the liquid layer inside the nucleus. We have a darker structure called the nucleolus. This is where we're going to make ribosomes. And then we have an envelope, the nuclear envelope shown here, And we're going to zoom in on the nuclear envelope and look at it more carefully in the next few slides. The nuclear envelope is really interesting because it's a double membrane. It's made of 2 phospholipid bilayers. There's more protection here than even in the regular cell membrane. So here, this is what a regular cell membrane looks like. And the nuclear envelope has 2 layers, so double protection. It separates DNA from the cytoplasm, and it also separates these processes, transcription and translation. Why is that? Remember, transcription is when we read DNA and we make messenger RNA, and that's going to happen inside the nucleus of eukaryotic cells because DNA is found inside the nucleus. After we make our RNA, it's going to leave through these nuclear pores, go into the cytoplasm where we are going to translate the messenger RNA into our polypeptide chain. Remember our chain of amino acids, and these will eventually become proteins. There are pores, like I mentioned. So these yellow structure are pores that perforate or make holes in this membrane, and that allows the nucleoplasm to be connected to the cytoplasm, and small things can flow back and forth. But if it's a larger molecule, there needs to be a set of steps and something called the nuclear localization signal in order for these larger molecules to go in or out of the nucleus. That string-like material that was in the nucleus is our DNA wrapped around histone proteins, the complex of which we call chromatin. So chromatin is DNA wrapped around or organized around these histone proteins that you find in eukaryotic cells. So most of the time we see the string-like structure, the chromatin, found inside the nucleus. But right before cells are about to divide, they condense even further into structures we call chromosomes. So here is a picture of a chromosome and an identical copy attached to it at the center. And we'll learn more about this in the genetics component. And I can see many types of chromosomes here on the right. So looking at this nucleus, I see chromatin, what we were just talking about, the loose DNA that is wrapped around histone proteins but not condensed into chromosomes yet because the cell's not dividing. Further inside the nucleus, we have the nucleolus, which I mentioned earlier, are where ribosomes are going to be assembled or produced. And this is by combining RNA and proteins. And it's a special type of RNA called ribosomal RNA or rRNA. There are regions within the DNA that will encode ribosomal RNA, and that will be combined with proteins to form the ribosomal subunits that will be exported out of the nucleus and eventually produce ribosomes. We'll see ribosomes are going to be these red structures, and some of them will be bound to a membrane, and then some of them will just be floating freely in the cytoplasm of the cell. So these ribosomes were produced in the nucleolus within the nucleus. What do they do? So they actually function to produce proteins. They put amino acids together to synthesize proteins. If I look at them more closely, I can see that they are made of ribosomal RNA and proteins, like we mentioned in the previous slide. They're found in both eukaryotes and prokaryotes, but they're bigger in eukaryotes. And if I examine them closely, I can see they're made of 2 subunits, a smaller subunit and a larger subunit. This red piece and this white piece was the smaller one. We're going to see later on how they assemble amino acids into proteins, but for now, I just want you to know that they produce proteins. You'll see ribosomes in 2 main places inside the cell. One is a set of free ribosomes that are just floating around inside the cytoplasm, and their job is to make proteins that are going to stay inside the cell. So these usually stay inside the cytoplasm and perform functions there. There will also be attached ribosomes that are attached to some kind of structure, and these are going to be proteins that are going to be exported, or they're going to be inside some kind of other membrane structure, something like a vesicle. Vesicle. Here I can kind of see some of those free ribosomes that are floating around in the cytoplasm. And I also see a bunch of those red ribosomes that are attached to a membrane. All right, our next organelle is the mitochondrion. This is singular. Mitochondria is plural. And these are shown here. These little structures that look like they have invaginations in the center. And here's another one shown here. Mitochondria are important because they produce energy for our cells in the form of something called ATP, or adenosine triphosphate. Mitochondria are interesting because inside the mitochondria, they actually have their own DNA and their own ribosomes. They are also comprised of two membranes like the nuclear envelope. They have an outer phospholipid bilayer and an inner phospholipid bilayer. And the inner membrane is folded up. Those folds are called cristae. And the liquid inside the mitochondria is called the mitochondrial matrix. So if I were to sketch this out, I would have an outer membrane and the inner membrane is folded up. And it kind of looks like that. That would be the cristae. And I see these memes kind of floating around the internet a lot. And I heard high school bio teachers use this one a lot, where they say roses are red, it's hot like hell, mitochondria are the powerhouse of the cell. And I like how the colors are shown here. Green is the outer membrane. Yellow, we have our inner membrane with the cristae. There's DNA and ribosomes floating around inside. And the inside, again, is called the matrix of the mitochondria. And I don't know if you've ever seen this one before, but it's pretty interesting that it says, Mom, you gave me something Dad never could. So during fertilization of the egg by the sperm, the egg cells, so human egg cells, are really big compared to a male sperm cell. That would be really small. So fertilization, what happens is the sperm cell basically injects or donates its nuclear information, its DNA, into the egg cell. But if you look at who donates most of the cytoplasm to the future baby, it's really the egg. The egg contains the mitochondria, the cytoplasm. So most of us get our mitochondria and our mitochondrial DNA from mom's egg cell. The sperm do contain mitochondria, but usually the mitochondria from sperm cells are destroyed during the fertilization process. Peroxisomes are another organelle found in eukaryotic cells, and they're usually small, round, and they look like a generic vesicle. They are surrounded by a single lipid bilayer, so just a single membrane layer, kind of like the plasma membrane. What happens here is they detoxify poisons, and this usually occurs through oxidation reactions. They can also use oxidation reactions to break down old fatty acids or old amino acids that need to be recycled or removed from the cell. Usually one byproduct that's generated because of these oxidation reactions is hydrogen peroxide, H2O2. And this is pretty damaging to cells and cell membranes, but as long as it's inside, as long as these reactions are happening inside of the peroxisome and hydrogen peroxide is inside the peroxisome, peroxisomes usually have enzymes like catalase that actually convert the hydrogen peroxide into water and oxygen gas, so it does not harm our cells. Vesicles and vacuoles in animal cells are membrane-bound sacs, and this would be a single phospholipid bilayer that store and transport things. So often you'll see they look like generic packets, generic circles or spheres inside the cell, and they can actually even add to or take away from the plasma membrane. So when something is taken into the cell, for example, an invagination of the membrane will form a vesicle, or if products inside a vesicle need to be released outside of the cell, the vesicle can fuse its membrane with the plasma membrane to add to it. So what are some other differences between animal and plant cells? Earlier I mentioned that plant cells look more rigid because of their cell wall. So they have that cell wall. They also have chloroplasts, which are organelles that allow them to go through photosynthesis, whereas humans and other animals. Unfortunately, we can't photosynthesize, although it would be awesome if I could. I would just lay out in the sun and build energy that way. Plant cells also have other plastids. Plastids are small organelles that contain pigments for photosynthesis or some kind of food. And they usually have a really big central vacuole that holds water, wastes, and nutrients. And animal cells don't have this large central vacuole. Animal cells have something known as a centrosome, and we also have organelles called lysosomes, but plant cells in general do not have that. And both animal and plant cells have structures called microtubule organizing centers, or MTOCs, but animal cells have something called centrioles that are associated with the microtubule organizing center, and these centrioles are organized into a location or a structure called the centrosome, and plant cells do not have this. And I can see some of those differences here. I can see that animal cells have a centrosome that plants don't have. They have lysosomes. Where can I find one here? Lysosomes. Here, the pink. Those contain digestive enzymes. Plant cells do not have that. Animal cells, I don't see a cell wall, so there's no cell wall. There's no central vacuole. And again, the centrosome is where that microtubule organizing center is found in both plant and animal cells, except plants don't have the centrosome structure or the centrioles that make up the centrosome. So here I have my plant cell. I see the cell wall. I see green color, so these chloroplasts. These are the organelles that allow for photosynthesis. I see plastids around and that large central vacuole that animal cells do not have. And again, plant cells don't have that centrosome. No centrosomes, no centrioles. So again, the centrosome is a microtubule organizing center, and it's found in animal cells, but not plant cells. In animal cells, it's usually found pretty close to the nucleus. A centrosome is made of two centrioles, or a pair of centrioles, that are perpendicular to each other. So they're lying perpendicular to each other. Each centriole, if I look carefully, here's one centriole, and here's the other one. Each centriole is made of 9 triplets of microtubules. So microtubules are these orange straw-like structures. These are actually proteins. And I see 9 triplets produce a centriole. And each of these triplets is connected to one another by the green, flat, non-tubulin proteins shown here. Their main purpose is to align and pull chromosomes apart during cell division. So when the cell is about to divide, all of the chromosomes are duplicated, and then they're pulled apart. So they're going to be separated into their 2 daughter cells. Lysosomes, they look like generic vesicles, again, inside the cell, but they have a very specific function. Sometimes we call them the garbage disposal of the cell. And again, these are found in animal cells, but not plant cells. In plant cells, usually they store their old stuff in vacuoles within the center of that plant cell. So lysosomes contain digestive enzymes, and the inside of the lysosome is also acidic compared to the rest of the cell. The combination of the digestive enzymes and acidic environment allow lysosomes to break down old proteins, sugars, lipids, nucleic acids, and even old organelles that the cell no longer needs, so that the pieces can be recycled and used for something else. Interestingly, if a cell needs to destroy itself through a process called apoptosis, apoptosis means programmed cell death. Sometimes it's called cell suicide. If a cell needs to destroy itself, what it does is basically break open all of its lysosomes. So if all of the lysosomes broke open all at once, the inside of the cell would have a much more acidic environment, and all of the digestive enzymes would be activated, basically chewing up the internal parts of the cell, and eventually the cell will die. So apoptosis sounds kind of crazy, but there are many instances where it's supposed to happen during normal cell development. One example is where it's not happening when it's supposed to is in the case of syndactyly, where you have a fusion, this webbing between adjacent fingers. So usually what happens is during pregnancy, the baby, or during gestation, the baby has webbed fingers. Babies have webbed fingers inside the uterus. And during the 7th or 8th week, apoptosis of this tissue should happen. When it doesn't happen, then we have syndactyly in these babies. All right, so now let's look at plants. Unlike animal cells, plants do have cell walls, and that's a pretty rigid structure found outside of their cell membrane, and you can see them under even the simplest microscopes in onions and other plants. They make the cell look really rigid. These cell walls are different from prokaryotes like bacteria because in plants, most of the time the cell walls are made of polysaccharide cellulose, which is a bunch of glucose linked together in those beta-1,4 linkages that humans could not digest. But in prokaryotes like bacteria, remember, their cell walls were made of peptidoglycan. If you guys are a fan of raw vegetables and you like the crunchiness of the raw vegetable like carrots or celery when you bite down, that crunchiness that you feel and you can hear is you tearing down the cell walls of our plants. And you don't really see this for cooked plants because when you're using the high heat, you break down the cell walls. Chloroplasts are only found in plants, but interestingly, they're similar in their structure to mitochondria because they're also double membrane. They have 2 phospholipid bilayers, and they also have their own DNA and ribosomes. So they have that outer membrane, and they have an inner membrane, and the liquid inside is called the stroma. That would be synonymous to the mitochondrial matrix. If I look inside, it doesn't show it in this picture, but the inner membrane is continuous with these stacks, these membranous stacks called thylakoids. So each sac is called a thylakoid, and a stack of them is called grana, or singular granum. These have photosynthetic pigments embedded within their membrane, such as chlorophyll, and they give it the chlorophyll gives these stacks a natural green color that absorbs light to go through photosynthesis, and we'll talk about that in a later chapter. So in plant cells, but not animal cells, we usually find a large central vacuole where they store water, nutrients, and sometimes even wastes. And it really helps regulate the water concentration inside the plant cell compared to their external environment. So if you ever have flowers or other vegetables or plants that wilt if you forget to water it, that's because the water has left the vacuole and you need to add more water and have water move into the vacuole for it to become firm again. And we'll be doing an experiment like this in the future where we take plants like celery and we add it to different conditions of water, salt water, regular water, deionized water, and we'll see what happens to the vacuole, if it fills with water or if it loses even more water, and if the celery becomes crispy and firm or if it wilts even further. So why do some organelles like mitochondria and chloroplasts have 2 membranes, and why do they have their own DNA and ribosomes? The idea behind this is endosymbiosis, or the endosymbiotic theory, which came about from Lynn Margulis. She was an American evolutionary biologist who passed away in 2011, actually. She's pretty famous as well because she was formerly married to the astronomer Carl Sagan. So what happens in endosymbiosis is we know that prokaryotic cells are the oldest type of cell. So we had this older, larger prokaryotic cell, and we think that these older cells engulfed smaller prokaryotic cells, perhaps a photosynthetic prokaryote, and that could have resulted in the formation of chloroplasts. And it could have also engulfed aerobic prokaryotes, which eventually results in the formation of mitochondria. That That would explain why these organelles have two membranes, the outer membrane being the larger cell's membrane and the inner membrane being the smaller prokaryotic cell's membrane. And it would also explain why these organelles have their own DNA and ribosomes. Again, this is just a theory, but we'll talk more about this in later chapters. That takes us to the end of part 2, our second video. In our third video, we'll look at the components and functions of something known as the endomembrane system.